In this lecture, we are going to do some exercises of the t-test, and in the previous lectures, we had discussed that what are the different types of the t-test and what are their specific requirements and what are their common requirements. And there are basically two types of the t-test that we studied. The student's t-test, which is the t-test for the independent means or the unmatched or the unpaired observations, and we studied the paired t-test, which is the t-test for dependent means or the matched observations. And we saw their general requirements, that what are the requirements for what are the requirements for these t-tests to be performed. And then we saw that how these two types of the t-tests they are executed, and we solved a couple of exercises over there for each of the tests. And we also discussed that how these t-tests can be performed on Microsoft Excel. So today we are going to do some further exercises so that you have a clear concept and differentiation of the two types of the t-test that we perform, that what are the conditions under which we have to perform the t-test for the independent means, and what are the conditions under which we have to perform the t-test for dependent means. So here we are with the exercise number 1 for today, and this exercise is about the plasma ACE levels. And ACE is the angiotensin converting enzyme, which is a very important enzyme found in animals. And this enzyme is involved in is part of the renin-angiotensin system, and this is involved in certain metabolic functions of the body, including the kidney functions and the blood pressure regulation. So this is a very important enzyme, and this is the exercise about the plasma ACE levels that were measured among healthy and hypertensive mice. And the unit of their measurement is picogram per deciliter here. So find out if there is a significant difference in the mean plasma ACE levels among healthy and hypertensive mice. So what kind of the t-test are we going to do? This is obviously we are going to apply a t-test because this is the comparison of mean values or the comparison of averages. So the question is that what kind of t-test are we going to use? Are we going to use the matched observations t-test or the unmatched observations t-test? So in this case we have two groups of mice, healthy mice and hypertensive mice, and the variable is the plasma ACE levels, and this variable is measured only once from each of the units in the two groups. So therefore these observations are independent, and we are going to apply the student's t-test, which is the t-test for independent means. So this is the data. These are the observations that we have. So these are the plasma ACE levels, and the green boxes or the green columns, they show the plasma ACE levels in healthy mice, and the blue boxes or the blue columns, they show the plasma ACE levels in the hypertensive mice. And these pink boxes, they show the number of mice, right? So this is just the serial number of mice. So here you can see that this is one group of mice. So in this group of mice we have 20 mice, and we have taken 20 observations. So one observation from each mouse. And what are these observations? These are the plasma ACE levels. So this constitutes one group, and these are 20 healthy mice. Now the next group is of the hypertensive mice, and these mice are also 20 in number, and we have 20 observations from these mice of the same variable, which is the plasma ACE levels, right? So I think the concept is clear to you, that what is the question and what kind of data do we have. Now this is the descriptive statistics for this analysis, for this exercise. So the number of healthy mice is 20, and the number of hypertensive mice is also 20. And the mean plasma ACE levels in healthy mice are 15.76 picograms per deciliter, and the plasma ACE levels in the hypertensive mice are 20.23 picograms per deciliter. And then the standard deviation of the plasma ACE levels from the mean value in the healthy mice is 2.669 picograms per deciliter, and in the hypertensive mice this is 3.2677 picograms per deciliter. And then in the healthy mice the variance is 7.1225 picograms per deciliter, and in the hypertensive mice this is 10.678 picograms per deciliter. So this is the descriptive statistics. Now what you have to do is that you have to assign numbers to these samples yourself. So I haven't assigned the numbers, that what is sample number 1 and what is sample number 2. So you have to assign these numbers yourself, that what you are going to label as sample number 1 and what you're going to label as sample number 2. So these are the two samples, or these are the two groups that you have. So this is our first exercise. Now we have the second exercise, and this exercise is about the same variable, and which is the plasma ACE levels. So in this case, the plasma ACE levels were measured among hypertensive mice before and after treatment with a drug. So in this case, we have only one type of the mice, one group of the mice, which are hypertensive mice. But in this case, again, the observations are different. So these observations are taken twice from each of these mouse. And these observations are taken before the treatment and after the treatment with a drug. So these are the matched observations. The observations are dependent, and these observations are present in pairs, and a pair of observations is taken from each mouse. So these are repeated measures. So we are going to apply the t-test for for matched observations in this case. So the question is, find out if there is a significant difference between the mean plasma ACE levels of mice before and after treatment of the drug. So this is our state of affairs. Now these are the observations that we have, and in the green boxes we have the observations of the plasma ACE levels before treatment, and in the blue boxes we have the plasma ACE levels after treatment. And in the pink boxes we have the serial number of mice. So you can see that there are 40 observations, but these observations are taken from 20 mice, which means that from each mouse we are getting a pair of observations, and observations are being repeatedly measured. So 15.4 and 8.4 is the first pair of observation that we have from mouse number 1, and so on. So we have a pair of observations from each mouse. So this is these are first 10 mice, the pairs of observations from the first 10 mice, and these are the pairs of observations from the next 10 mice. But are these two types of the mice, are these two separate groups? No, they are a single group, right? They are a single group, but the observations are repeated. The observations are matched, and these are the repeated measures observation from the same group of mice, right? So we are going to apply the t-test for the matched observations here. Next we have the exercise, which is about a loud explosion and its effect on the hearing loss. So we know that loud explosions, they can cause acute hearing loss in individuals. So in this case, a loud explosion occurred in a town, and hearing loss was measured among individuals living in a town affected by an explosion. And measurements were taken from individuals living within 0 to 1 kilometers radius of the site of explosion, and those living within 1 to 2 kilometers radius of the site of explosion. So now we have two groups of individuals, so we have to find out if the degree of hearing loss is same in individuals from both groups. So in this case we have two groups of individuals, two different groups of individuals. The observations are not repeated, the observations are independent, and one group of individuals are those who are close to the site of explosion, and the other group of individuals are those who are slightly away from the site of explosion. So we have these two groups, and we want to see that if the hearing loss is same within these two groups of individuals or no. So we are going to apply the t-test for independent means, because these are independent observations and these are taken from two different groups. These are unpaired or unmatched observations. And here we are with the observations, and in the green boxes or green columns we have the observations taken from individuals living within 1 kilometers radius of the site of explosion, and in the blue boxes we have the hearing loss measurements taken from individuals who are living in within 1 to 2 kilometers radius of the site of explosion. And in the pink boxes we have the number of individuals, the serial number of individuals. So here you can see that we have 50 individuals from group number 1 So we have 50 observations from group number 1, and these are those individuals who are living within 1 kilometer's radius of the site of explosion. And then we have another 50 observations, and these are from the individuals of the second group and individuals living within 2 kilometers radius of the site of explosion. So we have 100 observations, and these 100 observations, they are into two groups. They are from two groups, and each group is having 50 observations. Right. So this is the data that we have, and we have to apply what kind of test? We have to apply the t-test for the unpaired or unmatched observations in this case. So here we are with the descriptive statistics. So we have the first column for individuals within 1 kilometers, and we have the second column for individuals within 2 kilometers. So the number of individuals that were taken observations from in the first group are the 50 individuals, and the same number of individuals are from group number 2. And the mean hearing loss is 50 decibels from individuals living within 1 kilometer radius of the site of explosion, and the mean hearing loss value is 31 decibels for individuals living within 1 to 2 kilometers of the site of explosion. And then the standard deviation is 4.8354 decibels for individuals living within 1 kilometers and 5.9616 decibels for individuals living within 1 to 2 kilometers radius. And then the variance values, and the variance values are 23.3813 for individuals living within 1 kilometers radius and 35.5408 decibels for individuals living within 1 to 2 kilometers radius. So here we are with the descriptive statistics, and now we have to apply the t-test for independent means on these observations. So our exercise number 4 is also about the same scenario in which the effects of the loud explosion on hearing of individuals is being observed. So hearing loss was measured among individuals living in a town affected by a loud explosion, and measurements were taken from both ears of individuals living within 0 to 1 kilometers radius of the site of explosion, and find out if the degree of hearing loss is symmetrical among both ears of individuals or no. So in this case we have one group of individuals, but the observations are repeated, and the observations are paired observations, to be precise, that two observations are taken from each individual. So these individuals, all of them, they live within 0 to 1 kilometers radius of the site of explosion, and they constitute a single group, and the observations are present in pairs. So a pair of observation is taken from each individual, so therefore this data qualifies for the t-test for dependent means, or the t-test for the paired or the matched observations. So we are going to apply this t-test on this data. And here we are with the observations. So in the green columns of the green boxes, we have observations taken from the right ear, and in the blue boxes or the blue columns, we have the observations taken from the left ear. And in the pink boxes, we have the number of individuals. So you can see that there are 50 observations, 100 observations, and these observations are taken from 50 individuals. And these observations are paired. So 39 and 38 decibel is one pair of observation taken from individual number 1 from both ears of the individual. So here we are with the first 17 individuals, the paired observations taken from 17 individuals. These are the paired observations taken from another 17 individuals, and these are the paired observations taken from 16 individuals. So are these three different groups? No, these are not different groups. These are same group. So this is a single group, and we are going to apply the t-test for matched observations, because the observations are obviously different. The observations are paired. The observations, they are from the right ear and the left ear. So now I'm not going to solve the exercises in this tutorial. Why? Because you don't do it yourself, I know. So you have to do the exercises yourself, and I'm going to send you the link for Google Forms, and you have to upload your results onto that form, and you have to answer simple questions that will be asked on that Google form. So you have to do the exercises yourself, and you have to share the results with me. And I'm not going to give the details here right now, but in the class, when we have the question answer session, we are going to discuss the details, that how the exercise was solved and what are the correct answers. But you have to do this exercise yourself, and then we are going to discuss this exercise, right? So for every exercise, what you have to do is, and you have to formulate a research question for that, right? So you have these state of affairs with you, and you have to formulate research question for each of these exercises, and then you have to define the type of t-test to be performed and why, right? So you have to give the reasons that why you're going to perform this type of the t-test. And then you have to state the null and alternative hypotheses for your t-test. And then you have to apply f-test if applicable, and you have to apply f-test with proper protocol, by stating proper null hypothesis and by stating proper result, right? So you have to apply the f-test as well if it is applicable. And you obviously have to calculate the t-statistic. You have to define the degree of freedom, that what is the degree of freedom for your specific exercise. And in the end, you have to document your results. So write down your results in a scientific format. So this is what you have to do for each of the exercises, and you don't have to solve these exercises on, or you don't have to type them on the Word document. You can just solve them on paper and take the pictures of that paper and send it to me. But the thing is that you have to do it. You have to do these exercises so that you are prepared for your exams. Okay, and these exercises are also important so that you have a complete understanding of the concept, the principle behind each kind of the statistical analysis that you do. Alright, so this is it for today, and this lecture is not lengthy, and the reason is because you have to solve the exercises. So come prepared in the class with these solved exercises.